From Microbe TV, this is Matters Microbial, a podcast about the wonders of microbiology, microbiologists, and microbial centrism. This episode was recorded on February 22nd, 2024. Hello, Micronauts, and welcome once again to today's Quality Quorum. I'm your host, Dr. Mark Martin, Associate Professor of Biology at the University of Puget Sound in Tacoma, Washington. Today, believe it or not, is the 30th episode of Matters Microbial. I am very grateful to all of you who watch or listen to this podcast. Every guest on this podcast receives a packet of thank you items, including this small cross between yours truly and a tardigrade, which my students have long called a tardidoc. Which brings me to a wonderful Wunderkammer. These large standing tardidocs, which can be used to hold pencils, pens, inoculating loops, or plate spreaders. Perhaps we should have a listener giveaway for some of these at some point. My wife, Dr. Jennifer Quinn, and a friend designed this great hybrid. From time to time, I get messages about the podcast, and I'm grateful for each of them. One listener recently suggested a podcast session about some of the little-known historical figures in microbiology, like Annie Hesse, who first suggested using agar to solidify microbiological media. Believe it or not, some early microbiologists streaked out bacteria to single colonies on slices of potato. There is a link to the story of Annie Hesse in the show notes. This week in my introductory cell and molecular biology laboratory session, we ran a portion of last week's 16S ribosomal RNA PCR reactions on an agarose gel. And as you can see, many students had successful results. Those large bands, amplicons, are the clone genes. They cleaned up their reactions, and we have sent them out for DNA sequencing. Which means soon we will know the identities of their water bottle buddies, which is very exciting. Okay, to me. Following the Tiny Earth guidelines, students have been plating out bacteria from soil samples, looking for evidence of antimicrobial action. In this photograph, there are several candidates you can see that appear to inhibit other bacteria. We'll be analyzing them in the coming weeks. Ditto for moss samples is seen here. The lichen samples are not quite ready for analysis, but I look forward to them. Our goal, again, is to look for student-isolated bacteria that are producing antimicrobial compounds, perhaps new antibiotics. This is a real problem with the rise of antibiotic-resistant pathogens that needs to be addressed. Why not crowdsource it using students to teach them basic science, basic research, and basic observations? The Tiny Earth program, founded by Jo Handelsman and her colleagues, is described in the show notes, and I highly recommend it. During the last podcast, we explored the microbial denizens of caverns. That was fascinating. Today, we'll discuss the microbes that live deep within the sediment at the bottom of the ocean and elsewhere. As always, the microbes that exist in such environments have fascinating adaptation to those environments. So, it is a personal joy for me to introduce Dr. Jennifer Biddle of the School of Marine Science and Policy at the University of Delaware to this podcast to share her, with apologies to Jack Handy on Saturday Night Live, deep microbial thoughts. Welcome to our Quality Quorum, Jen. It's wonderful to see you again.
It's great to see you, and thanks so much for having me.
Oh, it's such a delight. You were on my list anyway. It's because, well, that laugh proves why. So the first thing that a lot of people would like to know a little bit about is how you got to where you are, because I know a little bit about that, and it's interesting.
Yeah, so how I got to where I am, I'm currently a professor at the University of Delaware, and I'm in the School of Marine Science and Policy. That journey started when I was probably 7 or 8, and when I put a wet slice of bread on the windowsill and waited for fungi to grow on it. I ruined my mom's windowsill, but she has forgiven me since, and realized that microbes were fun. When I got into high school, I loved DNA. And as I explored the concept of a genome and what DNA was able to give us, I realized that microbes have all the cool DNA. And so that was really my entry into microbiology. I did my degree at Rutgers University with fantastic microbiologists there, worked for a bit, and then went to graduate school at Penn State and have slowly walked out. So my PhD is actually in biochemistry and microbiology. And then I walked into geosciences and marine sciences, and I realized that the environment holds so many fascinating microbes, including, initially it was extremophiles I was interested in, and now just interested in all microbes in every environment and how they move around and how they grow and who they are.
I believe that you've had some interactions and worked with some quite famous people. So that's one reason because I had lectures from some of those people when I took, like, for example, the microbial diversity course at Woods Hole. And I can't imagine working with those people. It must have been very exciting.
Yeah, I've worked with some super fantastic people, and including, you mentioned Jo Handelsman in the show open, and I even tried to interview with her for a postdoc. She was very generous and very gracious, but it was actually when I sat down to try to write a postdoc fellowship proposal with her that I realized that the thing she was working on wasn't really what inspired me. And as I sat there and thought, if I had to write a proposal, what would it be? And that's when I emailed Andreas Teska, who's at UNC Chapel Hill, and said, Andreas, I want a postdoc with you. And everything, worked out brilliantly at the end. So I have to thank Jo for giving me the inspiration to realize I didn't want to go into soil and that I wanted to stay in the marine environment. So,
well, if you'll forgive me, we can both appreciate the sediment. So that's a wonderful story. And I know as part of what you do, you've done a lot of things at sea. Do you want to talk about that just a little bit?
Yes, I'd like to tell people I very much tried to sterilize the Gulf of Mexico. When I was at my postdoc position, we went on lots of cruises where we did lots of water filtration. And I really do feel like I must have put all of the water in the Gulf of Mexico through a filter at some point. Surprisingly, though, there are still microbes there. But we have been able to go out and collect many different kinds of samples, especially deep sea samples. We've been able to use ROVs, remote operated vehicles, to collect those in sediment tubes. I've been able to use an HOV, a human operated vehicle, to collect those. And so I think we'll talk about my time in HOV Alvin, and including using samples from the US and foreign drill ships in terms of getting really deep sediment samples through a process called scientific drilling. I've published on samples collected by divers. So it does take some effort to get down into the sediments. What is really fun is lately my graduate students have been walking in to get their samples. So we're able to work in some waist deep water where we just walk in and collect the samples.
So I think this is wonderful, but a lot of folks are going to be asking to themselves, it's the same issue when one talks about biofilms. And I've had people kind of dismiss biofilms, the uninitiated, not ones who've listened to the true microbial faith as you and I do. They'll call it like scum or things of that nature, when in fact, biofilms are extremely important. So when you say the word sediment, what does that mean to our listeners and viewers?
That's a great question, actually, because I think some people use the term in different ways. So what I think of as sediment is material that has fallen through a water column. So whether it's a shallow water column or a deep water column, it's a collection of, terrigenous material from the land. It's a collection of the bodies of things that lived in the water column. It's a collection of, chemical species that precipitate, in a marine environment and become all of this stuff at the bottom. So some people do sort of call soils sediments, but it really depends on how they're deposited. And so what I really think about sediments is that they're deposited in a depth layer so that the oldest stuff is at the bottom and then you have younger stuff on top. And basically there's a history that's included within them. And a lot of the sediments we work on, especially from the deep drilling sediments, are used to do things like recreate global ocean circulation models, determine what previous temperatures were in eras back to the Jurassic, And so things like that. So the microbes are living within this earth history, which is, I think, one of the fun parts of dealing with that.
It's such an interesting idea. And I mentioned at the very beginning that organisms adapt themselves to different conditions. And it's easy for someone who doesn't know a lot about microbial ecology to forget that even what you and I would call very small distances can be quite different in terms of energy availability, in terms of, and I'm not going to use oxygen. Can I rant for just a second with
Go
you?
for it.
So I have something I teach called the 3 deadly centricities, and it's a big deal in education to me. The first is certainly our introductory biology courses are relentlessly eukaryocentric. We focus solely on eukaryotes, which is not the majority of organisms on the planet. Secondly, and this is relevant to you and I, oxycentrism, because we're so focused on the use of oxygen. It's so funny when I tell my students it's just a place to, dump electrons, they look at me like, really? And I said, yeah, hold your breath. That pain, that's too many electrons. They don't like that. But the third thing, and this is really important to me, and this is why the diversity course meant so much to me, is what I call colicentrism, the idea that all microbes are like E. coli. So now we're talking about what some might say a layer, and I understand that there are very deep layers of sediment, but just imagine a layer of sediment like this. The top is very different from the bottom, and the middle is different from either. So what kinds of things do you find within sediment?
Oh my goodness. Well, that's a very big question. How much time do we have? So part of it depends on what kind of sediment there is. And let me just start with shallow stuff, right? So if you walk into a local lake or a river, you're going to be dealing with usually a fluffy layer at the top, which is young stuff. In that young stuff, you can actually have things like big chunks of plant material. You can have things like oysters and mussels growing into it. You can have things like worms and nematodes and things that bioturbate, right? And I do agree with your oxycentrism. I actually just told my class today about how bacteria have linear chromosomes, and they were like, "What?" And I was like, "Yeah, a couple of them do." But for this class, mostly it's going to be a big circle. But
I know.
um, so when you have those bioturbators, they can actually mess with the sediment layers. What is nice is that those bioturbators typically don't get very deep. And so there's a big question out there in the literature about what is the deep biosphere. I do believe most of the times I've defined it as greater than 1 meter below surface, but it depends where you are. For example, we've published on a local river that's only 2 miles from my house, and we actually did the age dating of the sediment material. What we found was that under 6 centimeters, the sediment got much, much, much older. So we went from having sediment that was within a 30-day time frame to under 6 centimeters being over hundreds of years old. And so you're able to sort of use some different proxies to determine how old things are and if a bioturbator might have been around. In terms of as you get deeper, I still have to publish this. We have an unpublished study that actually talks about, we created our own sediment column. So one of my graduate students, Kristen, was very ingenious, and she actually piped in some river water and started creating her own sediment. And we actually have this wonderful study that looks at which microbes respond best. And it's interesting. Some of them sort of die off quickly, right? They might like that initial fluffy carbon at the top, but as the sediment builds up, they can't handle it. Other ones seem to have some areas where they burst in activity. And so there's some that sort of seem to like it, when it gets anoxic, when maybe competition slows down, they start to do well. So they're sort of the middle of the sediment column. And then there were other ones that really wanted to get deeper before they started blooming. And so we're able to sort of divide things into, the opportunists that can't hang after a while versus the ones that are in their little niche specializations. And that all depends on which specialization it is. We've published on some sulfate methane transition zones where there's a lot of anaerobic methane oxidation. We've also published on nitrate transition zones where there's actually a lot of ammonia processing. And so depending on which, opportunity is there for that niche, different organisms will grow. And then there's others that need sort of these more stable chemistries to take off. One of the exciting things, which I shouldn't talk about because I have to actually get this publication out, but I have presented on it a bit, is we actually have a new phylum that we found. And what's really cool, I know I should have told you this beforehand, right? So there's a new phylum we found that only starts taking off under 50 meters below seafloor. So imagine 50 meters, it's a long distance. These microbes start to increase in abundance in really deep sediments. And so they really had to wait until to get into that right chemical condition to start growing. Um, so that's an exciting thing in terms of, basically it depends where you are. In the very general sense, we can talk about, in a global sense, what controls microbes. There's a theory out there that some of my colleagues have published that organic carbon is really the ultimate control. And I will say that seems to be the big factor, right? If there's a lot of organic carbon around, lots of things can grow, including your sedimentary archaea. And so if your viewers haven't heard it yet, right, archaea are a lot like bacteria. They're single-celled creatures, but their genetics, the way they work is just extremely different. Their membranes are different. And so we're still poking into that. And what's kind of frustrating is we've published on a site off of Costa Rica recently where the organic carbon isn't very different, but the bacteria and the archaea switch who's winning based on where they are. So it seems to be there might be a combination of sort of how fast the organic carbon can get to be anoxic. And depending on that might control sort of if archaea versus bacteria can win. What's also really frustrating is we're still sorting the genomics from those samples, and there doesn't seem to be any genomic clue about why one should win versus another. Big question about who lives in sediment really depends where you are, what depth you're at, and what could be around you. But of course, major controls as always is organic carbon and the available electron acceptors.
It's really important, I think, I talk about beginning micronauts and more advanced micronauts that are involved with this podcast. And I know that I didn't understand this when I was an undergraduate for quite a while until it was inserted into my brain by Sid Rittenberg. The idea is a simple one, that it's all about where the electrons go. And I think there's a quote, I'm going to have to look it up, that all of biochemistry is really an electron looking for a place to sit.
Mm-hmm.
And though that sounds kind of simple, there's a lot of truth to it. And I've tried to explain it to students. If you look at the energy potential from the NADH that we extract from our food, that we eventually dump the electrons carried by it onto oxygen, that's a huge energy differential. And we're used to that giving us quite a bit of, quote, ATP. But if you think about just making your living from the equivalent of glycolysis, other types of substrate level biochemistry, what you discover is that even in anaerobic communities that don't have that oxygen, there's a lot going on.
There is. And I think Jeff Gralnick started this, was hashtag SLP for life, right? Is this idea of substrate level phosphorylation. When there's not that much competition around, it actually gives you a lot of energy. And one thing that kind of still blows my mind, right, is if you think about how the human system works, and we are just fancy bacteria, right? And so, when we think about aerobic exercise versus anaerobic exercise, and even this is how sharks swim, right? So sharks have different white muscles versus red muscles, and that, fast swimmers basically shut off using oxygen, right? They switch to anaerobic metabolisms when they actually want to get energy faster. It's completely counterintuitive. But the TCA cycle takes a really long time. And it's something that I would love to figure out how to prove. And admittedly, I email Daniel Bond once or twice a year being like, Daniel, we have to figure this out. What's going on? Is there really a huge benefit to never having to worry about a terminal electron acceptor and passing your electrons different ways? And so it is something that I spend way too much time thinking about, because one of the big conundrums in deep biosphere is that there doesn't seem to be that much energy available. So there's microbial greats like Bo Jurgensen, and he published a lot on this idea that sulfate reduction should sort of be the ultimate end all be all of electrons in most sediments, which totally makes sense, right? I would want to put my electrons on sulfate if I was living in sediment. But yet again, when we started looking at it, and especially sort of in my PhD thesis, it turns out that fermentation is such a powerful force, right? And that's just carbon to carbon electrons, acceptors, and donors. And so it's kind of counterintuitive, this idea that the majority of life in the subsurface, even though chemically, thermodynamically, it makes sense, yeah, sure, push everything to sulfate, there might be a lot going on the way to eventually getting to sulfate. And I'm still not sure we actually can understand the amount of energy in the system, because all of our models predict that it shouldn't contain as many cells as it does. And so, yeah, so how are we powering all the cells in an energy limited environment is still a big question.
No, I'm loving this philosophically, because just as a lot of times we look at the behavior of another animal and we put human motives onto it, we anthropomorphize them. I think in a lot of ways when we look at microbes, we do the same thing. And it makes me cringe a little bit because we don't really have a microbial perspective. And I mean, you can say maybe I do, but even I don't. The point I'm trying to get at is that they have an agenda, and it's not to grow in a petri dish. It's not to grow in a 2059 tube. It's not to be in pure culture, that's for sure. So the point that I'm trying to get at is that we miss a lot of stuff because of our perceptions. So rather than anthropomorphizing, coli-morphizing, I don't know, oxymorphizing, but I think that that's true. So I'm getting way too philosophical. I think this is fabulous stuff. I'd like to talk a little bit to the listeners and viewers about archaea with you, since you're an archaeal aficionado.
Mm-hmm.
And the fact of the matter is, when you run into people, when I try to introduce them to the idea of archaea, they try to tell me that they're all extremophiles. Of course, they're not. And what's really interesting about it, I think, and you certainly can disagree with me, I'm nobody important, but I think that we didn't have something like E. coli among the archaea that to kind of shape our thinking. So archaea, as you mentioned again, are generally speaking single-celled organisms. They might appear to be bacteria because of their size and shape for the most part, but in fact, they're really have an interesting ancestry when you start to look at the DNA. Correct me if I'm wrong, but I believe the organization of RNA polymerase is much more similar to eukaryotes like ourselves.
Yeah, so most of transcription looks a lot more eukaryotic in the archaeal realm.
Mm-hmm. Right. And I hope later in a future podcast to talk a little bit about eukaryogenesis and the idea that the fusion between bacteria and archaea made what you and I call eukaryotes. But it's I think also counterintuitive, because most archaea are quite difficult to grow. And the ones that we can grow tend to be extremophiles. And I think that has influenced the way people think. So archaea are everywhere. Can you tell the listeners and viewers kind of what archaea do in the environment?
Yeah, well, so yeah, archaea are everywhere. And the idea that they were extremophiles is a very outdated idea. And so they do lots of things in the environment. A lot of the ones in the ocean do nitrogen processing. And so one of the hallmarks is that they tend to grow fairly slowly, and they tend to, so they're difficult to grow in culture because of that. But they tend to have a very streamlined metabolism where they're not doing a ton of things. Today in class, I called Pseudomonas the mom purse of microbial genomes, right? That's like, do you need a Band-Aid? Do you need a stopwatch? I've got it in my purse, don't worry. And the archaea in the ocean aren't as ubiquitous in terms of their abilities for that. In the sediments, a lot of the archaea do the processing of a lot of big organic compounds. So what's kind of interesting is in the sediments, when they're anaerobic, they're doing a lot of sugar processing, protein processing. They're doing lipid processing and beta oxidation. And so they tend to actually keep more abilities around. One thing that we have been noticing lately is that sometimes they have microbe or genome encoded abilities that seem to outweigh their total metabolism. So what do I mean by that? I mean that they might be breaking something down that's really big, but they might not have anywhere to put it. So what's interesting is we've published a couple of papers on how we find these archaea living with specific bacteria. So the bacteria seem to be the one who get past the metabolite, and the bacteria kind of finish things off. And so that is an interesting phenomenon in that, yes, some archaea can grow on their own just fine, but other ones seem to need a bacterial or even another archaeal partner to kind of do the work. And so they seem to be organized a little bit differently. But I will say, actually, we don't know how many bacteria actually really need buddies either yet. So maybe it's just easier to see because they're archaea. But
And that's interesting because, and I've talked about this with my students many times, the focus in microbiology for so long was on single cells, single colonies, pure cultures, when that occurs very seldom in nature, obviously. So were we studying the wrong thing? Yes and no. No, because there were things that needed addressing having to do with human health, and that was a good way to do that. But does that reflect how microbes live in nature? I would say no. So I've seen beautiful photographs where they use different types of vital dyes. You can show the archaeal partner on the outside of, for lack of a better term, this pellet with the bacteria inside or vice versa. So this idea, perhaps there's more of a partnership basis going on, is one I hold dear.
yeah, I would totally agree with that. And I'm laughing because when I was in graduate school, I started doing some cultivations from these deep sediments. And this was a environment that hadn't really been studied by cultivation yet at that point in time. And I had this anaerobic culture that I got down to about 8 different morphotypes, right? I could look through the microscope and see that there seemed to be 8 different kinds of bacteria in there. Well, maybe they were archaea, who knows? But I just remember my graduate advisor at the time was a very traditional microbiologist. And she kept saying, if it's not pure, if you can't get it pure, is it worth looking at? And I eventually abandoned the project. And now I'm kind of crying inside that I probably had this beautiful system that we could have studied this, partnership and symbiosis and really studied things in a community, but it wasn't appreciated at the time. And so that's long gone. I should maybe actually try to recreate that in my own lab. But that was
in my mind, I keep hearing the song "Happy Together." You know what I mean? Because it's really true. And it makes sense. I mean, we're a collection of different cells doing different things in collaboration. How is this really different?
Yeah, I've ended a lot of my talks with this idea of, especially for the deep biosphere, right? And there's a fantastic phrase. I don't actually know who started the phrase, but if you want to go fast, go alone. But if you want to go far, go together. And I actually use it. Maybe the company Peloton can sponsor your podcast eventually. But I talk about this idea of like, this is sort of what happened during the pandemic. Peloton got popular. Everyone wanted to do these things together. And so this concept of if you want to go far, you go together, right? And this is how bike riders, do long races. And this is maybe how microbes live in the deep biosphere.
And that's very different than a laboratory. I'm very interested in how cultures change, even starting from a single colony, and they'll change over cultivation. And I mean genetically change. And I'm not even going into Rich Lenski territory with over many, many thousands of generations. I think variability is really the key to all of this, and it's fascinating to me. But again, not the kind of things that people originally studied. I think you and I have a lot of ideas in our head that we should have pursued earlier. And we have to deal with that. And that's fine.
Yeah, I wasn't as brave as Rich Lenski to start an experiment without the promise of funding and continuation. So I'm very thankful to pioneers like him that, didn't listen maybe to their initial mentors or advisors to take the easier routes out. So
no, it's important to kind of stick by your guns in some ways. But I'm not going to second guess any choices that people make because I'm not that wise. But what I did want to say is that we've talked a couple of times on this podcast about why the average listener and viewer should really care about this work. What I want to do before we go into kind of the nitty-gritty of several projects that you've been doing, you've alluded to some of them already, but can you tell me a little bit, again, repeating for emphasis what you said, why the average person should care about those sediments? And I believe you started by talking them as a surrogate for changes in climate or changes in nutrient availability?
Yeah, and actually a lot of these deep sediments are actually really important, and I should say the microbes in the deep sediments are very important for global biogeochemical cycling. And so if we just think about methane, for example, there's a ton of methane created by Earth. It's also created by microbes, but it'll come up through the marine sediments. And you might be thinking like, oh no, methane is so bad in the climate. Well, over 90% of it gets eaten before it ever escapes the sediments, right? So you should be thankful to those microbes in the sediments. They're keeping things good for the rest of the earth. But
Mhm.
also just thinking about big processes in sulfur, processes in nitrogen, right? Almost every major element on our planet goes through a form of transformation in the sediments. And so that I think is really important. I also like to think about the idea, that as Earth was getting going, we know, so it's talking about, before oxygen. So in that time before oxygen, we know that there was a ton of bombardment. There were a lot of meteors hitting the Earth, right? And that Earth was potentially sterilized multiple times in terms of the oceans evaporating. And it's something that really captured my mind in graduate school was this idea that sediment might have been a refugia where these organisms could have continued living, right? So as the water starts to cool and come back, the sediment can help and reinoculate the rest of Earth and maybe have, restarted life a couple of times. It this kind of makes sense, right? That a lot of the archaea we see in these sediments are pointed at as perhaps the nearest partners to what we know became a eukaryote. And so when you do that eukaryogenesis episode, I think it'll be really fun to talk about that and think about why we find these organisms in these places, right? And that they might have been really key in how life got going on Earth.
Oh, I love the idea, but I'm a propeller head from way back. So, and I did you not get some funding from NASA?
Yeah, I've been funded a couple of times, and one of them was to look for the eukaryote type archaea in sediments. So
Yeah, and that's a good thing for I think listeners and viewers to know. When people talk about and I have a student who's interested in astrobiology. I'm old enough to remember when we called it exobiology, eek. But the thing is that she said to me, "So what do you study?" And I said, "There's a lot of microbiology, especially under unusual conditions. I won't even ask you if you think that there's existing life underneath the surface of Mars. I do. And if it is, it's almost certainly Archean."
well, and I will challenge you with what my graduate advisor, so my co-advisor Chris House at Penn State, I thought I was coming up with some great ideas. And the one thing that always stuck me was how do you make the nitrogen cycle work on Mars?
Yes,
And I still haven't
that
figured
is
that out.
no. At that, and the only thing I can say in response, and I won't take up any more time with it, and it comes back to again to that centrism idea that I talk about. I call it the n=1 problem. And if you only know of one type of way that life happened, does that mean there's no other way? You can't really say that. Maybe it's true, but you don't have evidence for that. So I always worry, how do you define it? And so that's what I'm thinking about there. But that's a subject, obviously, for another show. But I love this stuff. You can see it animates me literally. So that's a lot of fun. Jen, I've been reading a little bit about this possibility that one of the currents in the Atlantic Ocean might shut down. And there's been a lot of talk about how apocalyptic that might be. But relevant to your talk with us today, how could that affect, if this happens, issues of sediment formation? And may I add a second question? What do sediment issues, what can they tell us about climate change?
Yeah. And so I'll try to give you a somewhat short answer. So one of the cool things about sediments is that where they wind up isn't always where they started. And so when we think about currents, right, we have some massive fast currents, especially on the East Coast, our Gulf Current. And so it actually pushes some sediments up, basically off of like Newfoundland, in the area that the Titanic sank. Actually, there was a great expedition that looked at these basically deposits that get knocked up against structures in the ocean. So imagine, sort of a mountain at the bottom of the sea. They'll actually collect deposits next to them, and make basically a record of what was happening, but it might not have started where you find it, if that makes sense. What's really cool with that is I mentioned the ages of sediments. You can actually find really old things, really shallow. One of my favorite sites is actually a very old site off the coast of Brazil, where basically the modern sediments have been scoured away, and you're left with really, really, really old sediments in a very shallow area. So that's really fun. In terms of the current shutting down, that is a concern. To be very quick, basically, oceanic currents set up at the surface because of wind, but in the depths because of changes in temperature and salinity. If we have a lot of fresh water going in, right, from the melting of Greenland, let's say, it does change the speed at which the currents might run. And so that is a huge concern, and something that, looking at these old sediments, and again, it's not my research, it's our paleoclimate colleagues that have looked at it, they've shown that sediments across Earth's history have sped up and slowed down over time. And of course, this controls the global climate cycle. You might appreciate, right, as a microbiologist, that the reason Europe is currently warm is because of the Gulf Current. And so if that changes speed, it could change the climate in most of Europe, right? And so that is of great concern. I can't really necessarily predict what's going to happen. I believe the movie The Day After Tomorrow predicted it, right? And that we'll have a sudden ice age. But it is really disconcerting. And I will note, just in terms of climate change, the deep ocean hasn't necessarily caught the signature of climate change yet. So while the surface ocean is warming, there is still a lot of cooling. So we don't think about necessarily what's going to happen, but eventually there's a tipping point, right? There's a tipping point where the warmer temperature gets deeper. And I mentioned before the methane that is in sediments, like one of the huge concerns is if we warm too quickly, can the microbes keep up? And that's actually something I want to look at in the future. But this idea of like, what is that tipping point? Is it 1 degree? Is it 2 degree? What can they adapt? You mentioned adaptation in sediment. What can they adapt to? They've been stuck in pretty cold temperatures for a long time. Can we actually get any to live in a warmer environment? There's still big open questions. I do like to sometimes tell my undergrads the sort of harrowing story about how there's an area off the coast of North Carolina called the Blake Ridge. There's a ton of methane coming out there. It's also an area that is going to be highly affected by the changing currents. If we warm the Blake Plateau up, is there going to be a huge explosion of methane? It's not, unheard of. I don't know how likely it would be, but it's something that definitely, causes me to stop and think once in a while about making sure I am thinking about climate change and actually reacting to it.
So I like the fact that you talked about why people should care about what's taking place in the sediment. So if you wouldn't mind, could you talk about one or two of your projects that you were really excited about?
Oh, only one or two. So I am very excited about this potentially novel phylum, right? One of the things that we found it through Metagenomics, and it was actually found by a summer intern to my lab. We have a research experience for undergrads project or program funded by NSF, and they come for 8 weeks in the summer. He was going to be doing a microscopy project, trying to basically figure out different ways that you could identify sulfur using organisms through the microscope. And 2 weeks into his internship, the microscope broke. So I said, well, we have this old data set and maybe you can do some bioinformatics because I'm so sorry, like the repair guy can't come out yet. And lo and behold, he found this new phylum. So that is just a fun story. And I'm very excited about that.
So can
Yeah.
Can I break in for just a second? And for listeners and viewers, metagenomics, and my understanding is that term was originally coined by Joe Handelsman, and I love saying that because I want people to know that. So it's where you look at all of the DNA in a particular environment and you reassemble all of the genomes present there by looking for overlapping segments. I see no way that this could have been done without real improvements in computer science. And when you do that, you're able to construct genomes of organisms that you don't even have the ability to grow. And that's really critical for this particular situation. And so what your undergraduate visitor did was discover a whole new clade of life as part of his summer project.
As part of his summer project. And what was kind of shocking is I actually had a visiting scientist come after he had left. And I asked her, I said, could you try to find this somewhere else? Surely this isn't a different sample, right? She's looked over hundreds of samples. We cannot find this organism anywhere else.
Oh my goodness.
And that is still, we can talk about the rare biosphere for another 2 hours if you want, but this idea, right, that there's lots of microbes in a very small number floating around somewhere. And so maybe this thing does exist somewhere else, but we have not found it. We
That's
spent
amazing.
a good year looking for it somewhere else. And right now it seems to be only living in this deep area off Costa Rica in the sediments.
Wow. Now, I always tell people when someone discovers something like that, it's really easy for someone to say, well, is that important? And was it Faraday who talked about, like, is a newborn child important too? Wasn't that the quote about that? That it's easy for someone to say, is that important? But, there are lots of things that turn out to be important we hadn't expected them to be. And to discover this whole new area of life that people have not seen before, that's a pretty big deal.
Yeah, and with everyone sequencing, right? There's a ton of information out there.
Mm-hmm.
And the big discoveries have lessened, right? We don't find big differences that often. We're finding small differences, right? So maybe a new genus, maybe a new species. But to find something really big, it was really exciting. And made me actually check my undergrad's data a couple of times to say, are you sure you did the math right? So it was really astounding. But on the other hand, it completely makes sense. This is an anaerobic bacterium and it loves hydrogen based on its genome. And so it doesn't seem to be happy. What's really interesting also, I will just mention, hydrogen is really hard to measure in the environment, especially with the drill because the gas is escaping as you're collecting it, right? But this thing is living on top of an area where there's earthquakes. So it's living above a fault zone. I could see you go, so we actually wonder if the hydrogen is being created by the tectonic activity and Earth itself might be fueling this microbe, which also might explain why it's hard to find somewhere else because its requirements might be really specific in terms of the type of environment it needs.
Well, now you know where to start looking. You just need to go find an active fault line and go dig down there and hope it doesn't turn into a sci-fi movie. This is wonderful. And I'm really happy you brought out REUs, Research Experiences for Undergraduates. National Science Foundation funds these in many places, including your own institution. I wrote one 1,000,000 years ago that was useful. So you fund students to come to your institution in the summer and they study things there. And it's a really wonderful, wonderful thing. And there was nothing like that when I was coming up, as it were. But I cannot emphasize the level of importance where a student discovers something for themselves. They found it out. Someone else could say, oh, it's not terribly important. That isn't the point. They found it. Too many times in education, we simply have a series of demonstrations that students do, and that has value. But when they found something out, that's really where the good stuff is.
And what was really exciting was, he said, I don't know, something's wrong with this one. It's not getting a label. And I was like, what are you talking about? It's not good. Okay, let's like look at it. And then it's like, well, things don't get labeled if they're really different. This might, and sort of showing him through that process was really fun. So
Do you know who Isaac Asimov was?
the name is familiar. Yeah.
He's an old science fiction writer, passed away many years ago, and he was full of little quips. Well, he's full of other stuff too, but that's another story. But what he said is that science doesn't progress, and very Asimovian, with some damp Greek emerging from a bathtub saying, Eureka, right? Instead, it advances when someone looks down and goes, that's weird, because that is what discovery is.
Yeah, exactly.
Mm-hmm. To see what other people may have seen, but to think what no one else has. And that's what it's all about. So I am applauding this particular research project.
Well, and I can tell you one other fun project, and I will mention that this one is unfunded, but it's a combination. So you mentioned the microbial diversity course at Woods Hole. I was able to just send one of my graduate students there. And at that course, you're able to do a research project. And so I had looked at a symbiont of oysters. So Christie spira, it's a huge spirochete. It still fascinates me. And then my student actually decided to look at the gut microbiome of skeleton shrimp. And hopefully we can put a picture up of a skeleton shrimp. They are the ugliest things until you look at them for a long time, and then they turn cute. So they are these little amphipods. They're used as environmental indicators sometimes of pollution. They're a key species sort of in the intermediate trophic levels of a coastal estuarine area. And we started looking at their microbiomes. And so when Malik came back from Woods Hole, I said, well, we should have these here. Let's spend some time looking at the Delaware ones. So we have now data sets from Massachusetts. We have data sets from Delaware. My REU student last summer spent a lot of time looking at them. What's really fun, again, is the more you look, the more you find out sometimes. We actually started sequencing the cytochrome oxidases of these shrimp because we think that we have species that have never been recorded in the Atlantic before. And so it's still in progress. I cannot promise anything yet, but we've agreed it'll be a chapter of Malik's thesis to talk about not only the gut microbiome. What's really fun is we can track it based on the gender, the sex of the shrimp, and the life stage. And then looking at this idea, does it vary across different species of the shrimp? And we'll probably be able to now add to the natural history of the area to say actually that there's way more abundant populations that hadn't been noted in all of the guidebooks that tells you which shrimp live locally. So that's been really fun. It's gotten me very far out of my comfort zone. As a marine scientist, I do tell people, and I told them before they hired me, I have 1.5 credits of marine science in my educational history. I took a class on hydrothermal vents at Rutgers. And so I have been learning a lot about amphipods and learning that it's actually very hard to get sequences from their mitochondria because mitochondria look a lot like bacteria. And so we've had some fits and starts in trying to get that to work. But that's been a really fun project to think about. And, it's just one of these things where if you just have continued discovery over time and then you sit back and look at it, you realize, oh my goodness, we're going to be able to write a really fun paper about this. And it might actually really change the way some people are thinking about the distribution of some of these organisms.
This is amazing. 2 different ways you've talked about finding out new things that were unexpected. And since you're the mom of a teenager, correct?
Yes.
So one time when I went to Woods Hole, I got invited to be on, I don't know how this happened. It was very nice of them to be on someone's PhD committee at Woods Hole. And so they flew me out. I'm all wrung out. I'm tired. I go to the Captain Kidd. You know the Captain Kidd. Yeah. And I sit down with a big steaming cup of black coffee and one of those awful hamburgers that are so good. You know what I'm talking about, right?
Oh, yeah.
And there's a girl of maybe 14 years old. And she comes over to me at the table and says, "Are you a scientist?" And I said, because I've always been a smart aleck, I like to think so, right? He says, "Can you come sit with my dad and me?" So I walk over and here's this very quiet man clearly her father, and she's doing all the talking. He's doing nothing. And she starts telling me about the work her father had done at Woods Hole. And then she says to me, just like this, Jen, have you ever discovered a new species? And then I said, no, I can't. He goes, my daddy's discovered 7 so far. And then he looked all embarrassed. And I said, sir, you need to take this moment, fold it up and put it in your pocket because you're going to want that in a few years. So what a wonderful experience you have for your students. And of course, for the rest of your colleagues for this. I mean, I'm so impressed.
Well, I'm glad these are all, essentially happy accidents, right? Of finding things as we were en route to somewhere else. And people have asked me why I stay in academics and why I like being a professor. And part of it is just making a life out of happy accidents. And,
yeah,
I worked in industry for a very short time and had managers who would say like, oh, you need to get this. We need this exact, thing. And I realized my brain didn't like to work that way. And so I do like the ability to explore what's new, explore what's interesting, and try to follow it up. So.
Oh, I think this is such a great series. And I too worked in biotech for 7 years. It was the best thing that ever happened is when I got laid off. No, I cannot emphasize that enough because it was, and again, when I say this is not a general statement. It was soul crushing for me.
Yes. And I agree. Some people do great at it. And I have friends that are loving their jobs and doing excellent. And I'm actually very pleased. I'm actually able to work with them, some of them now, as an advisor to their company, which makes things really fun because you just get to have all sorts of ideas and then decide which one to follow up on. But I agree. It wasn't as exciting. And I tell people all the time that when I first started that position, I was in research and development at a very small company and we had no windows. And part of the reason is that the company was so secretive and that we had to sign all these non-disclosure agreements that I couldn't talk about what I was doing. And I couldn't talk to my family. I couldn't talk to my friends. I would just go to work and come out and I wouldn't have even known if it was sunny or if it rained or I wouldn't know anything. And as I didn't like that job, I started applying for other jobs. And I actually, back in the day, there were classifieds, right? And I actually applied through the classifieds to be a technician at Princeton University. And when I walked in for the interview there, the lab at Princeton had ceiling to floor windows. And I looked at the PI and I said, what are we doing? I'm in. I want sunlight. And so as I told people, I just made a career choice based on sunlight alone. The lab also was very fun and I had a great time there. But, sometimes your decisions don't have to be super weighty and necessarily make sense to other people. Sometimes they just have to be what your soul needs at the time. And at that time, I just needed windows.
Well, at the risk of talking about the way Marcus Aurelius wrote his meditations and trying to be a Stoic, everyone's different with their journey. And so I want to make sure it doesn't sound as if I'm judging other choices that people make. Good Lord, I've made some very unusual choices in my life I wouldn't wish on anybody. But the point I'm trying to get at is finding that path is so important. And that's something I want listeners and viewers to see is all the different paths that we get to that are I don't know, they feed the soul for me. And if someone is selling, I don't know, outboard motors and they enjoy doing it and they're making money at it and they like it, that's great. But for me, there's nothing like streaking a plate. I've got to tell you, it's really true.
Yes, very true.
Jen, one of the things that I enjoyed the most about going to the microbial diversity course at Woods Hole is getting a chance to see some of the things that, I'd only seen in documentaries on PBS. One of them was the Alvin, the human-operated vehicle. And you've been in Alvin. Can you talk a little bit about it?
Yeah, so I've been able to go 2 times to the bottom of the ocean in it. And it is a fantastic experience where you get loaded up on the ship and then they drop you off. They close up the hatch and then you descend into the water column. It is really cool to go through that twilight zone where there's bioluminescence. And then you get down to the bottom and you hope that the lights and the batteries will work. And so you're able to go then motor around the bottom of the ocean. We were able to look at a lot of cold seeps and hydrothermal areas in those cruises. And it's a very tight quarters. I am not very large, so I actually didn't really mind it. I can actually stand up in Alvin, so I'm 5'3". It turns out that's about how big Alvin is. But you're sitting there with a colleague with your feet, sort of in each other's faces, and the driver, the pilot is driving. And so it takes 3 people, and you look out these tiny portholes, and there's cameras everywhere for you to look through and see what the person see on the other side. But it's a really, really cool experience.
I heard it gets quite cold.
It does, actually. So I think one of the worst parts about it, or what I found to be really bad, I didn't necessarily mind getting cold because we would bring, sweatshirts and blankets, and we had things like that. But you spend all this time in this deep metal sphere in the cold. Well, guess what happens? As you get back to the surface and you start to warm up, all of the water vapor that you've been breathing out and sweating gets back into condensation. So the worst to me part of the experience was as you hit the surface, all of a sudden it becomes so humid and just like clammy, and then you start warming up. And those are actually the worst times I had in terms of sickness, in terms of bouncing around at the top. Once you get below the waves, you're fine. But that was the unfortunate part. So
I remember looking at the hatch, telling myself that they would have to hit my head with a sledgehammer to fit me through that tiny hatch. But I'm so delighted you've had an opportunity to do it twice.
I'm sure you would have fit just fine. You might not have been able to stand up, but
it was like I said, it's so surreal to me to have seen these things on television, and you did it.
Yeah, and I have to say, I laugh because my roommates in college were marine science majors, and so we all took this hydrothermal vents course together. And I think I just told myself, this is something I want to do. And then lo and behold, it's something I actually got to do. So it's helpful, again, sometimes you don't need a really clear idea of what you want in the future, just sort of an idea of like, this seems cool, right? And then you can go and do it.
Well, thanks for sharing that story. As we start to kind of close out a little bit here, Jen, I like to ask visitors, what was the one observation or moment that sold you on microbiology? I mean, I know what it was for me. What is it for you?
I do remember. Well, I should tell you a funny story. When I was in high school, I had an environmental sciences teacher who, was teaching us about the diversity of DNA. And I think maybe that was really the place where I really got sparked. In traditional New Jersey story, he was arrested 3 years later for selling drugs to students. But so he made a difference in my life, at least in a good way. But then part of it also was I really wanted to be a graphic artist. But my graphic artist, the teacher in high school told me I was awful at graphic art. And so very ironically, my science teacher was very encouraging and the art teacher was very discouraging. And so that alone might have pushed me in the direction to get into microbiology.
Now, that's very interesting. That you sure that science teacher didn't go by the nickname of Heisenberg? When we meet, ask me some questions about some unusual things when I got laid off from biotech, and one unusual job in particular that I won't share with you now, but you'll enjoy the story.
I certainly will.
I wanted to thank you again for taking the time to chat with us. Sharing your deep microbial thoughts, a big deal to me. I've learned a lot about the sediment. I have things that I want to write to you about involving that situation with the Puget Sound here where I live and work. And I hope I can bend your ear a little bit on that. But I wanted to make sure to thank you for your time and my very, very best wishes to your colleagues and your family.
Thank you so much. And thank you so much for having me. I'm very happy to support you in doing this. And it was a lot of fun.
Thanks so much. This has been Matters Microbial, a weekly podcast about the wonders of our microbial world and the people who study it. You can send questions, suggestions, or comments to me at mattersmicrobial@gmail.com. Show notes from today's episode, as usual with tasty links, can be found at microbe.tv/mm. If you like our work, please consider supporting us at microbe.tv/contribute. I'm Doc Martin, and you can find me in the biology department of the University of Puget Sound in Tacoma, Washington. Dr. Jennifer Biddle is in the School of Marine Science and Policy at the University of Delaware. Many, many thanks, as always, to David Renata for superb editing, and Reber Clark, as always, for the wonderfully quirky music. I hope you've all enjoyed being part of our Quality Quorum for today. See you next time on Matters Microbial.